Fighting Blindness Canada's Viewpoint is a virtual education series that brings you the latest in vision research presented by health experts from across Canada. The webinar you're about to watch is a recording. To learn more about the research we fund and upcoming webinars and events, please visit our website at fightingblindness.ca. This Viewpoint webinar is proudly presented by Bayer and supported by AGTC, Allergan and Abbvie Company, and Novartis. We'd also like to thank Accessible Media Inc., our national media partner. Everyone here should download the AMI app, which is a fabulous resource. And you can watch excellent programming, including previous Fighting Blindness Canada events. Thank you for watching. We hope you enjoy this webinar and share your thoughts with us in the comment section below.
And welcome to Dr. Lefebvre, Dr. Krishnaswamy, and Dr. Ballios. I'm going to start by talking to Dr. Ballios, and then we'll get Dr. Lefebvre and Dr. Krishnaswamy on in a few minutes as well. Welcome, Dr. Ballios. We always love talking to you. So for people who don't know, Dr. Ballios was the first recipient of Fighting Blindness Canada's Clinician Scientist Emerging Leader Award, and it was awarded while he was doing his residency at University of Toronto, and please correct me if I'm wrong about any of this. And then after completing his fellowship in Harvard, Dr. Ballios has returned to Toronto and is now an assistant professor with support from the J. Ardith Hill FBC Professorship in Ocular Genetics, and is also a scientist at the Krembil and has a clinical practice at UHN and Sunnybrook. So that was a lot of things and a very, very full career and schedule, I'm sure. So I was wondering to start with, if you can tell us a little bit more about your research and clinical practice and how those two intersect.
Sure. Thanks, Larissa. Yeah, I can tell you that I'm very excited to be back in Toronto and working as a clinician scientist at a couple of different great Toronto institutions. My research focus is primarily on new approaches to retinal regeneration. And for a long time, I've been very interested in how we can use stem cells, not only to model and understand how retinal degeneration works in the plastic petri dish in a very controlled environment, but also how we could potentially design better therapeutics to regenerate the light-sensitive cells that are lost during retinal degeneration. And that includes both acquired disease, what we think of as conditions that are common, like age-related macular degeneration, but I also have a very, very special interest in inherited retinal disease and genetic retinal degeneration. It dovetails so nicely with my clinical practice, where I have a specialty in inherited and ocular genetic disease, and that's really the focus of much of my clinical practice. And so it's been wonderful to see patients from all across the province and across Canada who've been over the last few months coming to see me in my clinic and getting some information about what's going on with their eyes and beginning to understand how they might be able to connect with new research opportunities. So I think in this particular space of ocular genetic conditions, there's going to continue to be a very intimate relationship between what happens clinically and what patients see clinically, and what we're trying to do in the research sphere, and how we try to move those discoveries back to patients in the clinic. So I think talking about optogenetics today is very topical, as it's one of the new emerging, hopefully potentially new therapeutics for patients, and I'm sure we'll get into it.
Absolutely. And just before we do, I was just wondering, so what came first for you? An interest in ophthalmology, or an interest in inherited retinal diseases, or an interest in regenerative medicine?
Yeah, I think when I talk to people who work as physician scientists and who practice on both at the bench, in the laboratory, and also in the clinic, people often come to it in two different ways. Sometimes they become a clinician scientist because they had an innate love of medicine, and they wanted to work on some of the most challenging questions in medicine, and that led to their love of research. And vice versa, some clinician scientists had a love of research that led them to want to pursue a life in clinical work that helps a certain patient population. I would say I'm probably more of the latter. I have been interested in neuroscience and retinal development and retinal disease for a long time. And I did my PhD in retinal stem cell biology and became very interested in how the retina works. And I think as I continued to work on those very interesting scientific problems, I really fell in love with the clinical practice of ophthalmology and saw a great opportunity to help people who were in need. And that's how I came to life as a clinician scientist.
Thank you for sharing. So jumping into sort of the meat of this program, which I'm sure a lot of people want to talk about, is optogenetics, as you mentioned. So I was wondering if you could just start us off with a bit of a primer. What is optogenetics?
Sure. So optogenetics is a way of combining what we're all really excited about the best parts of gene therapy, which are about trying to take new genes and put them into the retina, to use the retina in different ways, but trying to get around the fact that in many retinal conditions, most of the innately light sensitive cells that we have when we're born, the rod and the cone photoreceptors that detect light, they go away slowly over time. And so optogenetics is an approach that says, let's use the best parts of the retina that continue to be healthy throughout our lives to get better and better vision as time goes on. And so to think about optogenetics, we have to take a little bit of a step back and think about retinal degenerative conditions in general. Most retinal degenerative conditions, whether they're acquired like age-related macular degeneration or whether they're inherited retinal diseases like retinitis pigmentosa, they involve a slow loss of light-sensitive cells in the retina. And our retina is not like other tissues in our body. It does not naturally regenerate these light-sensitive cells. We are born with only as many as we're going to get, and they slowly go away over time in these diseases. And the goal of traditional gene therapy, and it's a whole nother topic of conversation, but in very brief, the traditional goal of gene therapy is to support the health and function of the light-sensitive cells that you have left. And depending on how quickly you can get to them, in whatever condition you're talking about, you can try to intervene. In optogenetics, we're often targeting and thinking about those patients in which most of the light-sensitive cells have gone away, where there are not many of those rod and cone photoreceptors left. And the approach of optogenetics says, you know what, let's forget about those light-sensitive cells that are going away over time. And let's try to take those other cells in the retina. And what I tell patients they are is they're helper cells. They're cells that help the light-sensitive cells do their business, to communicate with the brain. And let's turn those other cells in the retina into light-sensitive cells themselves. And in that way, we can take advantage of those cells because they stick around until very late in life. And how can we make those cells light-sensitive? And optogenetics is all about the different strategies that scientists are taking to try to make those cells become light-sensitive cells themselves. And in that way, try to regenerate and restore vision.
That's great. So sort of a follow-up question that I've always wondered is for these other cells. So I understand that in many diseases, the photoreceptor cells or light-sensing cells get damaged and die. And so you're trying to turn the other cells that remain into light sensors. Are there any diseases or common diseases where those other light-sensing cells, those sort of in-between cells that are communicating between the photoreceptors and the brain, degenerate and die? Because I feel like we talk a lot about the photoreceptor cells, but not about the other ones.
Absolutely. So yes, there are other conditions where those cells are primarily the cells that are affected. And I think the best example, or the most common example, let's say, that most people would be able to relate to is a condition called glaucoma. And in glaucoma, one of the light-sensitive helper cells called the ganglion cell that helps to send the light signal from the retina to the brain, those cells slowly go away over time. And so these optogenetic approaches are not really typically being designed for conditions like glaucoma, where the helper cells are primarily the ones going away. Interestingly, in glaucoma, while those helper cells go away, often the light-sensitive cells, the photoreceptors, remain. So it's very interesting in retinal diseases and retinal conditions. there's often this one or the other kind of approach where there's some conditions that either affect the photoreceptors and leave the other helper cells alone, or they affect those helper cells like the ganglion cells like glaucoma, and they leave the photoreceptors alone. So optogenetics is all about trying to help that first group of conditions where the light sensitive photoreceptors are going away and you have those helper ganglion cells remain.
So I think you've sort of touched on this a few times now about who would benefit from a potential optogenetics therapy. So you mentioned that currently it would be more for individuals who have lost a lot of their light sensing cells. So if you still have a lot of light sensing cells available, it might not be the therapy for you. And also that it's for specifically for individuals who've lost the light sensing cells and not some of these other helper cells. So would this potential therapy work for all types of inherited retinal diseases, or would there be some that it would be more useful for than others?
Well, I think you touch on two important things, and I think that there is a lot of hope in optogenetics that this approach could be a general approach. If we think about inherited retinal disease as a group of diseases, we know that there's over 300 different individual genes that can cause inherited retinal disease. And so it's an incredibly heterogeneous and a diverse set of conditions that are caused by a number of different genes, and most people's genes are different. And so if you're thinking of a gene therapy to replace that misspelled gene or correct that misspelled gene inside a cell in the retina, you're going to need to design a unique therapy for every single one of those over 300 genes. The hope of optogenetics is that you can say, you know, let's stop thinking about all of those different genes important for vision. Let's just focus on one approach to regenerate vision loss. And so I think the hope is that regardless of which gene you have that causes your genetic retinal condition, optogenetics could be an approach that would be helpful for you as long as there were those helper cells remaining within your retina. And yes, I think the earliest application of optogenetics as it emerges as a therapy, hopefully as we see more and more data as the years go on, I think the earliest patients it's going to be tried in are those who've lost most of those light sensitive cells. Those are the patients we don't have as many options for potentially for treatment, for gene therapy. But if it's successful, I can imagine it being used at earlier and earlier stages in the disease if we find the right patients to use them in. So I think in the end of the day, it's all about trying to provide patients with genetic retinal disease more choice for what kind of therapies they could potentially have in the future, without forgetting about some of those patients who have genetic retinal disease whose gene may be a very rare gene that causes genetic retinal disease. You know, some of the genes that cause inherited retinal disease are very common. Genes like USH2A, EYS, you know, I'm throwing out the names of some of these genes, but there's a lot of patients who have misspellings of these genes causing their disease. Some of our patients have misspellings in genes that are, they may be one of only a handful of patients in the whole wide world who have misspellings in that gene. When will gene therapy get to them? It's not clear. Optogenetics may be an approach that's going to get to those needy patients sooner. And so that's another reason that makes it so exciting. And
you mentioned like sort of the difference between optogenetics and another therapy we talk about a lot, which is gene therapy. I was wondering if you could just explain a little bit about how optogenetics works. Like, how do you make non-light sensing cells light sensitive?
So to try to understand this, you have to go back to what makes a light sensitive cell light sensitive. And photoreceptors are like little machines, and within them they have proteins, and these proteins are like the parts of a car's engine. They do their work and they make the cell function. And some of those proteins are involved in the detection of light. They're light sensitive molecules that can absorb a photon of light that enters the eye and convert it into an electrical signal. And light sensitive photoreceptors make these proteins naturally, and the other helper cells in the retina, they don't make these proteins, so they don't detect light. The goal of optogenetics is to introduce that piece of DNA that makes that light sensitive protein into those helper cells. And so it uses many of the same technologies that gene therapy has been using. Gene therapy uses viruses to deliver the gene into the light-sensitive cells inside the eye. The virus is like the delivery truck. It's like the Amazon truck delivering the package, right? The precious cargo, which is the gene. And instead of using that virus to deliver a light-sensitive gene to a photoreceptor, we're using that virus to deliver a light-sensitive gene to one of these helper cells. And so we use the same approaches to designing these viruses, but instead of targeting the photoreceptors, we target these other helper cells like ganglion cells. So the virus delivers the gene, and the gene encodes something like a protein that detects light, like either a modified version of a protein called rhodopsin, or a modified version of a protein called cone-opsin, or even new custom designed light-sensitive molecules. And we try to get those into the ganglion cells and the other helper cells inside the retina. And by having those in those cells, they can suddenly detect light.
So you're using sort of a gene therapy technique, but it's instead of replacing a faulty gene, you're adding in a new gene altogether, if that's what I understand.
The same toolkit that we would use for gene therapy, just instead of targeting the photoreceptors as the cells we want to treat, we're targeting the helper cells.
That's great. And so you've already we've sort of keep talking about the word potential therapy and in the future. So I was wondering if you could maybe tell us a little bit about some optogenetic therapies that are either in development, or how soon do you think there will be optogenetic therapies available?
So there's already a few clinical trials going on of optogenetic therapies, and there have been early clinical studies even going back 5 to 10 years in optogenetic therapy. And one of the problems that's plagued optogenetics over the last decade has been trying to find the right light-sensitive protein to get into these cells. And it turns out that not all light-sensitive proteins are made equally, and they have to be able to have what we call fast kinetics. They have to be able to turn on and off very quickly as the light flashes and photons enter the eye. And they also have to have a really favorable, what we call dynamic range, which means they have to be able to detect light over a large range of illuminance, right? Because we don't all live our life in the same light setting with a very bright light, like high noon on a Sunday in a cloudless sky. You know, we want these molecules to work under low light conditions, under the lamplight, or various levels of illuminance. And most of the molecules that have been tried so far, they require very high levels of illuminance to stimulate them. And that's nice because you can stimulate them, but doesn't always result in the most useful vision. If you think about how we use our vision every day, we don't turn on every light in the room, and it can be hard to detect contrast and shades and parts of the vision that patients really need to have useful vision, let's say. And so over the last 5 years or so, we've seen a lot of new light-sensitive proteins being developed that have a better dynamic range, that are more useful over a number of different illuminances, and also have faster kinetics, right? They turn on and off much faster. And so some of the newer ongoing clinical trials make use of these newer molecules, optogenetic molecules, these newer light-sensitive molecules. And most of the studies right now are in the earliest stages, phase 1 and early phase 2 studies to see if they're going to be efficacious. And they've recruited patients with low vision, patients who aren't able to see the eye chart, and who may not even be able to detect any light at all because most of their light-sensitive cells have gone away. And there have been some exciting early reports from these trials suggesting that patients who were not able to detect light are now able to detect some level of illuminance in a room, and that's really exciting. I think these are the baby steps that the field needs to take. I think as these trials go on, we look forward to seeing more results in patients with better and better baseline vision, to see if we can see more sophisticated types of responses out of these studies. But it's already in clinical trials, and so these results will continue to come out as the next few years go by. So that's really encouraging, because a lot of therapies, especially in gene therapies for various rare genetic gene types, many of them haven't even gotten to clinical trials yet. So that's really exciting that already optogenetics, which could be a general approach for many patients, is there. So it's always hard to pin someone down and say, okay, Dr. Ballios, how long? To see the results in 6 years, we'll know if therapy works or not. But I can say that we're already in clinical trials, and we're going to see new results over the next 1, 2, and 3 years out of these studies. And we'll know very quickly whether these therapies are going to be successful, because the companies will decide whether they want to move to more advanced phases of studies, right? They'll announce in the next 1 or 2 years, things are looking good enough, we want to recruit for a phase 3 study. So I think that we're going to start to see soon what they can do with optogenetics.
Yeah, it's really exciting to start to see some of the early results come in, and like you're saying, potentially small results, but really exciting first steps in this field. Just as a last follow-up question, can you see ways that optogenic therapies could be combined potentially with other therapies, or just thinking about what you mentioned about how some of the proteins that are making cells light sensitive, they don't have a big range. Could you put in different types of proteins? So one that has that senses light at this range, and then another one that senses light at a different range, instead of just using one single protein?
Sure, I think, and scientists are already trying these approaches in the lab to design combinatorial optogenetic therapies, to use multiple different light-sensitive cells that have a better dynamic range over specific illuminances. Translating any of these new approaches into a first-in-human trial, it requires a lot of careful planning, a lot of investment, a lot of preclinical data and evidence for safety and efficacy. And so I do think that we'll see more of that over time. I think if we are going to see it, it will be spurred on by early successes in the more basic and standard optogenetic approaches that we're already seeing in clinical studies. So if they start to gain traction and show some success, just like any field, we'll start to see variations and permutations on this. But yes, absolutely, people are thinking about combinatorial therapies, even about targeting specific light-sensitive molecules to specific cell types within the retina, specific helper cells, either those ganglion cells or other helper cells within the retina. And different light-sensitive molecules in different cells may give better and better responses. But this is the exciting thing about being scientists, and you're going to hear more about from the other speakers. Trying different approaches in the lab is where all of this starts. So what we're seeing in clinical trials is the culmination of over a decade of work in optogenetics. So what you'll be hearing about in terms of new ways to stimulate the retina is the next generation of therapies that we're going to be seeing as the years unfold.
Thank you so much, Dr. Vallios, for giving us a great overview of optogenetics. And we're going to move over to Dr. Lefebvre and Dr. Krishnaswamy, and then we'll bring you back for a Q&A at the end. I'm sure there'll be lots of questions. So welcome to the two of you. And as a quick introduction, Dr. Lefebvre is a senior scientist at SickKids and an assistant professor at University of Toronto. And she also holds the Candor Research Chair in Developmental Neural Circuitry. And Dr. Arjun Krishnaswamy is an assistant professor at McGill University, who also has a focus on neural circuits. And we're really excited because they were just awarded a research grant in our 2021 research grant competition. And sitting through the review panels, I can tell you that the reviewers are really excited about this project. So we're so excited to be funding it. So as a starting point, I was wondering if the two of you could briefly tell us a bit about your sort of broader research interests and how you developed your interest in vision research and optogenetics. Maybe I'll start with you, Dr. Lefebvre.
Okay, great. Thank you, Larissa. And I'm really excited to be here and to tell you about our research, but also to learn more from your community. So I run a research lab at SickKids, and kind of the broad goal of our research is to study the development of the nervous system, in particular to understand how different types of nerve cells that have different jobs are generated during development and how they wire up in these very specific patterns. We study this in areas of the nervous system that are devoted to vision, so primarily in the retina, and then other areas, visual targets in the brain, such as the cortex. So the focus of my research has been primarily basic research. We're interested in gaining a fundamental understanding of how these different nerve cells arise during development, what are the genetic pathways that are responsible for generating different types of cells, and what are the molecular pathways that are important for kind of wiring them up. We study this in animal models, and we've been studying this in the inner retina. And as we'll elaborate below, the inner retina is a very kind of complex tissue with diverse population of retinal cells that have very important jobs, but also very specific wiring patterns that is essential for them to do their jobs. And so, we've been studying this at the level of molecules, at the level of single cells, and we've developed kind of really important developmental insights into the events that lead to the development and the function of the retina. And so more recently, we've been motivated to leverage this understanding of how the retina develops to study retinal diseases. And in parallel, we've also been developing tools to study how the retina develops and forms these connections. And then been thinking about, well, can we also use these tools to get a better understanding of how the wiring between the retinal cells change in disease? And then can we use these tools then to tell us, well, are these connections maintained and are they strong enough to be harnessed in a system where you want to reactivate them, where you want to make your helper cells into these light sensitive cells when photoreceptors are absent? And if we do that, can we kind of reinstate neural transmission within the retina that's important for vision? So that's been kind of the trajectory. Again, we were very much interested in studying the development of the retina, but then use that knowledge and use the tools that we make along the way to get some deep insights into what happens in retinal disease.
That's great. So really using sort of discovery biology to inform future research into sort of more translational aspects.
Yes.
And Dr. Krishnaswamy, how about you? Where did your interest in vision or neural circuits come from?
Yeah, and I mean, in a lot of ways, I have a very similar kind of entry point. As Julie described, we study the mouse retina and its connections with brain targets. And we do this in the lab because there's been a bunch of recent advances in this part of the nervous system that allow us to mark and monitor and manipulate cells, neurons in this region. So we can ask really basic questions about how neurons connect up to one another and how they form circuits, which is kind of how the brain computes, right? So how we would, in the visual system, how it sees, those kinds of things. So we can relate patterns of connections between neurons to what these neurons do en masse. We kind of ask questions about what genes are important for connecting different cells together to create these important patterns, and then what the consequences of some of these patterns are for the function of the circuit. So how does it detect particular things, or in some cases, more recently in the lab, how do the signals that it detects relate to an experimental model's ability to see, so kind of behavioral approaches. So these are the kinds of things we're into in the lab, and more recently, kind of via Julie, we've also gotten into a translational angle. There's been a lot of great work done on the basic science of circuits in the retina and elsewhere, which puts us in a great position to ask what happens in cases of degenerative diseases, such as we've been discussing, and other things.
That's great. And we're going to get sort of into the nitty gritty of circuits, but a question we often get is, how do scientists collaborate, and do scientists collaborate on the research? And I think this is a really great example of Julie and Arjun collaborating on this project. Julie's in Toronto, Arjun's in Montreal. How did this collaboration start? Have you had a long-term connection, or was it very specific to this project?
Both.
Both. I think it's classical collaborate. It's very synergistic collaboration in that we have complementary approaches and we study similar questions using different techniques or from different vantage points. So I would say we've been really focused on studying genetic and molecular mechanisms important for the development of the retina, but particularly looking at how this relates to the structural organization of the retina. And Arjun has taken on more functional approaches to study this at the level of the neural signals that these retinal connections transmit, and he can elaborate on that. But another thing is that we've at one point we worked together as scientists during our respective training. So we've known each other for a while. We were both postdoctoral fellows together in the same lab in the US. We both established our labs in Canada, and this we've had these ideas to collaborate for some time, and this was really a great opportunity to collaborate, to launch kind of the first translational projects in our lab and to work together.
That's great. Yeah. So, do you have anything to add to that?
No. Well said.
Yeah. So getting into circuits, both of you mentioned the idea of circuits, and I was wondering if you could just explain what you mean when you talk about circuits, and especially within the context of the retina.
Do you want me to do that, Julie?
Sure.
Okay. Right. So Brian kind of talked about these photosensitive, these light-sensitive cells in the retina. They're called photoreceptors. And then there are these other cells that we've been talking about, and I guess in this discussion, calling them helper cells. So the retina has a bunch of different kinds of neurons, and the signals that it sends to the brain isn't just, it's not like the picture you would snap on your phone camera. It's something of an interpretation of the scene that comes into the eyes. And so the way that interpretation comes about is it begins with the photoreceptors, the light-sensitive cells. So these are like the pixels in your phone camera. All of the circuits end with a kind of cell called a retinal ganglion cell, which converts the photoreceptor signal into some interpretation, and it sends it to the brain via the optic nerve. And that's kind of the early step of vision. The circuitry comes about when those ganglion cells connect to sort of an intervening layer of neurons. And those neurons relay signals from the photoreceptors down to the ganglion cells, or themselves kind of interpret those signals before sending them to the ganglion cells. And the ganglion cells sort of compare both ideas, both views of the photoreceptor signal, and then send the brain its analysis. And as it turns out, there's been sort of a bunch of work over the last 20, 30, maybe even more years. It turns out that those intervening layers, let's call them interneurons, they come in a variety of flavors. And the ganglion cells don't just hook up to them in a random fashion. They connect to really specific ones. And when they do that, they become selective for certain things. So in the human eye, we think about colors, kind of a color opponency, seeing reds over greens or greens over reds, those kinds of things. In the mouse retina, where we do a lot of our work, there are cells that sense motion in particular directions. So the brain kind of gets this signal, which isn't exactly an image of the world, but more a sort of interpretation of what's moving, what color are things, where are the edges, those kinds of things. And so that's what the circuits do.
So are you saying that there would be some, say, retinal ganglion cells that would interpret, say you were seeing a ball being thrown. So some of them would see the motion part of it, some of it would see the color, some of it would see the edges, and your brain would then interpret all these different signals into a ball being thrown.
Yes, so certainly that view of things is probably very true for the experimental model that we used. I mean, whether that happens in a human eye, there are differences, important differences between the two, is unclear. But I think that's a reasonable approximation. Yeah, I think that's a reasonable idea.
Okay. So going back to the questions, I'm sort of jumping around a little bit here. And so I think you've sort of discussed quite a bit, actually really nicely, about the retina and the different layers in the cell and how the different layers are responsible for different things. So I was wondering if you could maybe start talking about, in your particular research project, how are you using sort of this information, and what techniques are you trying to use to convert some of these retinal cells into light sensing cells? Oh, sorry, you're on mute, Julie.
I'll just start with a few, and then Arjun can also talk about his technologies, because this is where things are coming nicely. So we generated a toolkit that really harnesses the power of optogenetics. And so we've devised a gene therapy strategy for experimental model to deliver optogenetics, a channel rhodopsin, which is the opsin gene that gives a cell, any cell that expresses it, sensitivity to light, as Dr. Vallios explained. And this strategy allows us to really kind of control which cells within the retina express this opsin gene and becomes an optogenetically activatable neuron. And this is, of course, done in an experimental model system, lacking photoreceptors, and so it's a blinded retina. There's two aspects to this toolkit. So the first is there's a cell within the inner retina, a bipolar cell upstream within the circuit that is then acquires this ability to sense light. And then what we want to do, another part of the toolkit, is to then assess, well, which cells downstream of the circuit, because the circuit is a pathway of cells and their connections that transmit information. And so which cells downstream are able to be activated by this stimulation and by this neural transmission. And essentially this tool allows us to then kind of identify which cells are able to respond to the upstream neural signal. And it basically tells us that the cells are structurally connected and also functionally connected.
And Dr. Krishnaswamy, do you want to add anything about the techniques that you're using?
Yeah, no, I think that's a good summary of things. So to figure out between the model that Julie mentioned, where the retina lacks these photosensitive cells, and normal animals, we'd like to see whether the circuits that we just talked about, do they look the same? Are the ganglion cells we talked about, are they hooked up to the right guys upstream, so that if you were to make the upstream neurons photosensitive, so they become the new photoreceptors, does everything else work out the same way? Are the kinds of signals, these interpretations we discussed, are they going to look the same? These are the kinds of things the grant was interested in. Well, it's focused on.
And I guess what is from either your past data or from other research, what do you think is going to happen? So do you think those connections in those circuits are the same? Do you have a sort of a hypothesis or a guess?
Many people have looked at models in which where there's considerable photoreceptor degeneration, and what they see structurally is that the other cells of the inner retina, the other layers, remain intact. And so we think that there will be some degree of, some connections will be maintained. But the limitation of these past studies is they haven't looked at the resolution of wiring that we're both proposing, at the resolution of individual circuits. That we have a variety of tools where we can selectively activate or manipulate one circuit at a time and look to see whether or not the wiring is correct. Both, we can visually do that and also measure the neural signals. So, I think my hypothesis is that there will be some degree of circuitry that is maintained and can be harnessed and usable. But there might be some important changes, and I think this approach will give us that type of information.
that was probably an unfair question, but I was just sort of interested to know, and we're looking forward to hearing more about it.
Yeah. I mean, we can see that there's one circuit that we study quite a bit in our lab, and Arjun has made a number of important contributions, and these are circuits that are important for direction selectivity. And so the retina has this, in terms of their ability to sense different elements of visual information. One is that retinal ganglion cells are tuned to respond or to become activated when they see light in a particular motion. And this is very important for motion sensing. A lot of the cells that are in the circuits, they're present in these animal models. Their connections look okay. I'm not sure if they're optimal, and we're still doing the experiments, but it was quite amazing to see that these circuits that are very important in the model that we study, they're present, they're structurally intact. So we'll see how they compute and whether or not there's some synaptic changes.
Yeah, that's right. Yeah. A subset of those, just to give you a sense of the importance of those signals, a subset of those direction selective neurons are at least in mice, and the feeling is in other mammals too, are important for stabilizing our gaze. So they help stabilize the eye, because your head is always moving, the body's always moving, but the image never changes, right? And so they're an important part of that pathway. And so they're just one of the many circuits that we're interested in kind of seeing, whether they're normal.
And in terms of how you figure out if making something light sensitive, if the other circuits are working. So not obviously, but you mentioned that you will be looking at if other cells are activated. Are you also doing any more functional tests on the animals, or is that more of another type of project?
Yeah, well, you want to go?
No, you go ahead.
Right. So kind of some of the functional tests we're doing are at the level of individual neurons, these direction selective cells. There's another level at which we can kind of survey many ganglion cells, like hundreds and hundreds and hundreds of them. And we can show patterns and see whether the signals that come out of those cells look like those that would come out of a retina where the photoreceptors are still there. And then in one of our final things that we'd like to try is to take those exact same retinas, the ones where we've introduced the channel rhodopsin and made the bipolar cells light sensitive. We'd like to test them out on some simple behavioral tasks. So one would be this kind of reflexive eye movement, this gaze stabilization thing. There is a set of innate visual behaviors in mice that have recently been published that we'd like to try out. So animals don't really have to learn to do it, which is advantageous for us, but it requires vision. And so we're gonna see how things work out.
It's really interesting.
And I just wanted to add, so the techniques that I'm a big fan of Arjun's technique, because his idea and his technologies that he created in order to record from hundreds of different ganglion cells at once it's really cutting edge. It's a really kind of fantastic neuroscience approach to studying the function of the retina. And I think it's really, really well suited for these translational questions to understand or to identify the cell types within the retina that are particularly vulnerable, and then those that are resilient in a disease model. Because once you know those, once you can start categorizing who's okay and who needs help, you can probably focus on the circuits that are maintained or that are strong, and focus the optogenetic strategies on those.
Right.
On those circuits to, perhaps it'll be part of those baby steps, and it'll be an important step in kind of devising a good strategy and also an optogenetic strategy in order to give you a particular range of visual ability.
I think that's a really great segue to the next question, which was if the project is, don't necessarily want to say successful, but based on what the information you will learn from this project, what do you think the impact of that information could be on optogenetics therapies, of course, but other types of potential treatments.
Maybe I think one type of information that will be very important will be, again, to identify which retinal cells in the inner retina are resilient and they maintain their connectivity. That despite this loss of photoreceptors and this loss of light sensitivity, that they still have all their parts, they're still connected, they can still be driven if you put more gas into the car. And it may not be 100%, but I think kind of that framework will allow can then be used for a number of therapies. So whether you're restoring light sensation through stem cell technologies, transplantation technologies, etc., regardless of the therapy or the strategy you want to use to reinstate light sensitivity, you still need your downstream retina to be able to compute that visual information.
It gives us a measurement, Larissa. It gives us a measurement of, where things stand, like how normal these cells are, or how much like their regular counterparts they resemble. And as other optogenetic tools come online, stronger ones, faster ones, those kinds of things, we can then apply it in the same experiment and then measure again and see if we got improvement.
Mm.
Yeah, and so
Yeah.
And we can sort of measure that improvement in terms of the connectivity that Julie mentioned, in terms of how many of these cells reactivate, those kinds of things.
Mm-hmm. Thank you. I think it's really interesting, the idea of how important a tool is in research. So I think we all obviously always want to talk about the next treatment, but getting to that next treatment takes this discovery research, and it takes development of really important measurements and tools. And I think you described that really well. So thank you. The last question, just sort of more broadly about sort of, I guess it might fit more into your general research interests. What are there other diseases that optogenetics research or the optogenetics technique is being used in, or is it very specific for the retina?
Mm-hmm.
You or me?
Yeah, so you go ahead.
Yeah.
I can add a few things. Do you want to go ahead?
Oh, sure, yeah. In the basic sciences, it's being used everywhere. The ability to control neurons with light has like transformed the entire discipline. And the reasons are simple. You don't just stimulate neurons. So we often understand neurons in terms of what they say to the next neuron. And in the olden times, people would have to put implants like electrodes and things and stimulate, and that's bad for all kinds of reasons. But if you can do it non-invasively with light, you can study all kinds of stuff. And so people are using it. Here at McGill, there are people using it to study how pain receptors in the skin signal through cells in the spinal cord and give you that sense. There are folks who are interested in kind of stimulating cells deep in the brain involved in motor control and that kinds of thing. The scope is really enormous.
And the person who, there are several individuals who contributed to the development or the idea of optogenetics, of using this using an opsin from a algae, harness that ability to sense light and introduce it and use it for gene therapy. This has been kind of evolved and through a number of groups. But one particular individual who was key, Carl Diceroth, wrote a book. It was called, I think, Projections. And so he's a clinician scientist. He's an MD, PhD, a psychiatrist who works with patients with major depressive disorders. And, in this book kind of talks about how, as a neuroscientist, he really wants to try to understand, he was motivated to try to understand, what by the time I see these patients, there's so many problems and so many symptoms. And one really has to try to get a fundamental understanding of how the brain is wired, and then what are the key entry points. And I think that has kind of really shaped his research. And he created Optogenetics, which has really changed the face of neuroscience. And to this day, I think he and many others are thinking about how to use Optogenetics as a way to provide a treatment for a variety of CNS disorders, as Arjun described. So.
And final question, I promise, is so you mentioned the algae protein. Is there a reason that scientists are looking more at sort of algae proteins as opposed to using human opsins?
Well, you don't really want to use a human opsin. Well, one I think it was more strategic, right, Arjun? It's because then the algae opsins are
they're self-contained. So
yeah.
let me explain. So when a photoreceptor, which has its opsin, when it catches a photon, it has to use other proteins, 3 others, to turn it into an electrical signal. And there's advantages to doing that. But in an algae, it's all done in one protein. And so that means you can just borrow that gene, put it in the viruses that Brian mentioned earlier, and deliver them to cells, and it'll work just as you want it to. There's some trade-offs there, but I think that's sort of the major advantage. There are folks who are trying the idea you suggest, though.
That's great. I just had never known that, but that is a very clear reason. It's much easier to put one gene into a cell than three separate.
Yeah. Well, I think as molecular biologists, we've always used proteins made by other animals, other creatures, bacteria, etc. We kind of harness some of those tools and those proteins that no longer have that function in higher organisms, such as mammals or humans. But it gives us little tricks to manipulate or to bypass the machinery that exists in the mammalian cell, to manipulate and give a new direction or a new instruction to the nerve cell.
They're related, though. The two are both part of this opsin family, which is super old, like really, really, really old.
Well, thank you both. And I'm going to actually call Morgan back, and then we will get back to, we have to give you a little bit of a break, and then we'll get back with the Q&A in a few minutes.
Thank you so much to all of our speakers for all of that information. My brain is buzzing right now. It's a lot of great information. So we will give everyone just a moment to take a short break. And I notice we have tons of questions that are already coming in, which is fantastic. So do please add yours into the Q&A. But for just a moment, I'm going to introduce my colleague, Doug Smith. At Viewpoint this year, we're trying to highlight some different ways that you out there can also get involved with FBC. And Doug is going to talk to you just for a few moments about one of those ways. Over to you, Doug.
Thank you. We interrupt this regular scheduled program to share some exciting news. Hi, everyone. My name is Doug Smith, and I'm the manager of Plan Giving and Donor Relations here at Fighting Blindness Canada. First of all, I want to say a big thank you to those who are listening today who are supporters of Fighting Blindness Canada. We could not do what we do without your support. There are many ways to support Fighting Blindness Canada. Could be an annual gift, could be a monthly gift, or a leadership gift, just to name a few. So what's this exciting news that I've interrupted our regular scheduled program today? Did you know that many of our fellow Canadians are leaving a charity or charities in their will or estate plans after providing for their family needs? This is just another way to continue to fund Fighting Blindness Canada by considering a gift in your will. A gift from your assets of any size, when you no longer need them, will give you the power to fight blindness in the future. A gift in your will is a key part of the fight for sight, the power to change lives. Perhaps you have never thought of giving a gift in this way, or maybe you have already included Fighting Blindness Canada in your will. And if so, I say thank you so very much. Either way, I would love to have a conversation with you about leaving Fighting Blindness Canada in your will. So please feel free to reach out to me. An email will be sent out after this webinar with contact information included in it, so you know how to reach me. Thank you again for your support of Fighting Blindness Canada. We are so grateful to all of you. And now back to our regular scheduled program. Morgan, back to you.
Thank you so much, Doug. Always a pleasure. All right, so let us get into so many great questions. So there's a lot of questions, both that have come sort of in ahead of time and in our chat today, asking, if optogenetics could apply to a particular condition. So maybe I'll just go through them, and would love, if you know of any research in that area, or if it would be applicable. I know we have touched on a lot of this already, but just to sort of view that for people. So I had a question from Vivian about glaucoma and retinal detachment. And I think, Dr. Ballios, you touched a little bit about this, that it's not really necessarily a treatment for glaucoma, but potentially for the retinal issues.
Yeah, its applicability to any specific disease, I think, really depends on what changes with the anatomy in that disease. And in retinal detachment, that is one of those exceptional conditions where there can be changes both to the light-sensitive cells, even after the retina is reattached, but also to the cells that communicate, the helper cells I discussed earlier, the ganglion cells, and that communicate with the brain. And so when there is a condition that affects both cell types, this makes the hurdle much higher for therapies. But I think that ultimately therapies that approach both sides of the coin, regenerating light-sensitive cells and also improving how those cells communicate with the brain through approaches like optogenetics, could eventually be helpful. It may not be the first disease or condition that finds its way into clinical trials, but once therapies are approved from clinical trials, then ophthalmology and other medical specialties have a track record of trying different therapies for different medical indications. So once something's shown to be safe and effective for something, then it's shown to be safe, and ophthalmologists try to find out if it can be effective for other conditions. So even though there may not be a clinical trial for optogenetics and retinal detachment, I'm not pessimistic that those approaches may ultimately be helpful for those conditions in the end. We just don't know enough yet about whether that therapy will be helpful. So it's a bit of roundabout way of saying I don't know for that particular condition, but it's better to have more options than one option. Yeah.
Absolutely. I'm not familiar with this as CRAO. We have Tushar is saying that his wife lost some vision due to CRAO after an embolization procedure. Would therapy be applicable in that case?
So a central retinal artery occlusion, a CRAO, is a condition that affects the blood supply to the inner retina that was being discussed earlier. And in CRAO, usually it's those helper cells, those ganglion cells and bipolar cells and other cells in the inner retina that go away as time goes on after a CRAO. And so that presents a hurdle similar to conditions like glaucoma, where we need to figure out ways to regenerate the retina's ability to get the light, the signal from photoreceptors to the brain. So again, it's going to require some creativity to develop new techniques in that respect. It's not obvious that optogenetics itself would be applicable to CRAO, but every retinal condition is unique and every patient's presentation is unique. And it all depends on how many of those light sensitive and or helper cells are left in the retina after any particular disease insult, whether it be a CRAO or whether it be a retinal detachment. So I think some of the same principles apply.
Thank you. Yeah, and we always get inevitably with any of our viewpoints, everyone wants to know if it's a potential for their particular condition. So, but I think you make such a good point that not only is every condition different, every patient is different, and the way that that condition presents, adds layers to figuring it all out. So I have another question here from Edward. I think I'll pass over to Dr. Lefebvre and Dr. Krishnaswamy. So he's wondering if optogenetics will also be applicable to RPE cells. He says, my understanding is that new cones won't be useful if they are not RPE cells to support the new photoreceptors.
Yes, I think the strategy of optogenetics is best, in my imagination right now, for cells downstream of the photoreceptors. Because the RPE are, serve a very important role for supporting the integrity and the function of photoreceptors, but they're not transmitting neural information or neural activity. And so, and one of the unfortunate thing is whether the primary, if there's a genetic effect either on RPE or photoreceptors, they both kind of contribute to their malfunction. So they're quite interlinked. But I think optogenetics is best downstream. Arjun, I don't know if you have other thoughts on whether it could be.
Yeah, no, that's well put. Yeah.
Thank you. So I have another question here from Kim. I'm going to give this one to Dr. Ballios because Dr. Ballios is my go-to every time I have a stem cell question. So Kim wants to know, and this might not be fair, but which is more promising right now, optogenetics or stem cell treatments?
Wow, what a loaded question. For those of us doing stem cell research, it strikes right at the heart of what we do. I think of it as this. You may not like my answer, but I think of them as serving very different purposes and addressing very different goals for treatment. I continue to believe that stem cell therapy holds great promise for regenerating the light sensitive cells inside the retina for those people who've lost many or even the majority of their light sensitive cells. I'm glad that we are thinking about other techniques to regenerate vision for those patients where a lot of those light sensitive cells have gone away. I think they are parallel efforts to vision regeneration because they address different anatomical regions of the retina to try to regenerate vision. It's hard to compare them. This is a bit like comparing apples and oranges because they're trying to manipulate the visual pathway in very different ways. So I'm glad to see that people are working on both approaches because at the end of the day, whether one succeeds or the other succeeds, it's better to have more irons in the fire and more options for patients for conditions where we never had any treatments before for decades and decades, where we could make clinical diagnoses, but we had to tell patients just what to expect over time and vision loss. I think that it's good that both are moving forward. I don't see them as much of a competition between the two fields. They have very different goals, so it's hard to compare their outcomes at all. It's hard to imagine what you might say is more promising. I think they're both very promising methods of vision regeneration, and I'm glad that both are being worked on.
Very well said, thank you. We have another question here. This is considering the neural component between retina and vision. Is there any specific research around dementia and vision loss? Specifically, could vision loss begin in dementia before actual dementia symptoms or brain shrinkage occur? I don't know if anyone has any insights on that particular question.
I'm not sure we have the composition of panelists to address dementia research. You know, there is a whole nother layer to how the brain interprets visual information from the environment and then associates that information with other stimuli like touch and taste and smell. And this is a realm of neuro-ophthalmology and neuroscience and neurology. I think we understand that patients who have dementia can often have difficulty with interpreting visual stimuli in the context of their disease. But I'm not sure that the panelists today, I mean, at least myself, I only speak for myself, who think very specifically about the retina and retinal conditions can speak to what's going on in dementia and neuromodulatory research in that area. But I do know that a lot of my neurology colleagues who are interested in dementia and Alzheimer's disease are very interested in how to train and work with patients in vision rehabilitation to make things easier for them in their disease. There's some exciting research going on there. I don't think I have the expertise to speak about it in a very specific way.
Absolutely. Maybe that's a potential future viewpoint. We'll have to think about that. So lots of questions coming up, I noticed, in both the Q&A and the chat, and also through our email about clinical trials. And we can, again, you know, Dr. Lefebvre and Dr. Krishnaswamy were talking about the basic science, but then moving into human trials, people have questions, including, would there be an age restriction on the trial? If someone takes part in a clinical trial, does it render them ineligible to participate in other trials? And how important is genetic testing to these trials? So would someone like to just talk about clinical trials in general and how all those things fit in?
Do you want me to talk about clinical trials, Morgan?
Sure.
Very, very briefly, clinical trials are very, very picky about the profile of patients that they recruit into a study. And they do that with very good intentions, all the best of intentions, because clinical trials are just that. They're trying something new in people. And they're really experiments. When we're talking about new medications or first-in-class medications, we're talking about experiments in people. And experiments have to be very well controlled, which means that you're trying to recruit a group of people to the trial and enroll them who have very similar clinical features, so that you can use the power of having multiple observations to give you some idea of the trend of what's going on. Is the therapy safe? Is it effective? Is it regenerating vision in the right ways that we expect or we hope it to? So many trials restrict not only the age of patients, but they also make restrictions on the level of vision that patients have and the level of retinal function. So trials can be very, very specific. Many trials start with patients with lower levels of vision on the eye chart. Many of them typically start with adult patients and not pediatric patients. And so these are, you know, especially for medications that are first in class. In other words, a new gene, a new target, a new therapy that's being used for the first time in humans. A lot of caution is taken in introducing these therapies. So it can be very specific as to who gets enrolled, and that can be frustrating for patients who really want to participate in trials. But I think also it's important to recognize that once a trial is successful, if it is successful and a medication is approved for use, then that really opens the door to application for all sorts of patients. So that's encouraging too, and we all hope for the best results out of all of our trials.
And I'll just add, I'm not an expert in clinical trials. I'm not a clinician, but I just wanted to raise the point that there are a lot of biotech companies and a lot of companies that are kind of taking some of the discoveries from labs and trying to improve a variety of ways in which to either introduce genes of interest or to introduce these new technologies. And I think this is a very, very exciting time right now for startup companies and biotechnology companies. So for example, there are many companies that are really trying to focus on designing the best ways for gene therapy to deliver particular gene cargo or optogenetics to specific cell types within the retina. And they're testing a variety of viral vectors in order to really improve and to make the delivery as precise as possible so that the side effects or the measures of success are most optimal. So I think that's another very exciting space is the biotech and the growth in these technologies and bringing kind of some of the discoveries from labs, but also within these tech companies themselves. And another, in the case of optogenetics, it's wearable devices, because this will be an important aspect of optogenetics. There will have to be ways in which to deliver the light. And so there are a lot of engineering biotechs out there who are also trying to solve this problem.
Morgan, the last part of your question I just recalled was about genetic testing. Just as a plug for genetic testing, I really think that in the modern practice of ocular genetic disease, genetic testing is really important. It's the way in which we define how a genetic condition is affecting a person's retina. And it, beyond just telling us information about why the patient has disease, it adds two more really important pieces of information. Number two is, how did this happen within the family, and who else might be at risk for having this condition across multiple generations? So who else do we need to think about as potential people who might need help or treatment in the future? And number three is, many of the therapies that are being developed, even if it's not optogenetics, if it's gene-specific therapies, they depend on knowing that gene and that specific misspelling in the DNA. And it tells us so much information about what's going on in the retina that it opens up so many more potential doors and options for patients that I think if we suspect a patient has an ocular genetic condition, we should be encouraging, with proper counseling, we should be encouraging genetic testing in this day and age. It's not a perfect science. It doesn't always identify the misspelling in the gene, and that can be frustrating for patients and families. But when it does, it provides very valuable and useful information.
Absolutely. I do the health info line here at FBC, and it's one of the #1 questions I always get is about genetic testing and how do I get it and should I get it? So, if there are people out there that have more questions about that, I'm happy to help. You can email me at healthinfo@fightingblindness.ca. I've also just popped into the chat. In Canada, there was a few questions about getting genetic testing covered. It is covered by the government in all provinces. So I've put a link there that has guides on how you can get genetic testing in your province and start that conversation with your doctor about getting that treatment. I have a companion who has joined me. So I'm going to ask another question here. This one's from Milton. So Milton says, what kind of vision would a person have if all the ganglion cells make photosensitive proteins? For instance, the feature extraction that occurs by processing of information in the retina layers would not occur. Just a sec. Unless appropriate ganglion cells are transfected with appropriate color sensors, there'd be no color vision. Optogenetics is a great approach. It would be the appropriate to discuss the expected limitations of vision obtained through optogenetics. I think that might be for Dr. Lefebvre or Dr. Krishnaswamy. Could we tackle that one?
Arjun, do you want to go ahead? You might have some good insights.
Yeah, so that's a good question, and whoever asked the question has seen it correctly, that if you put in the sensors into all the ganglion cells and they all react the same way, then that isn't necessarily how the retina works normally. So hard to know what that means for vision, but certainly it would change the way the retina works. Now, I think the way around that, as Julie mentioned earlier, kind of in another question, is that to stimulate the ganglion cells, the light sensitive bit allows us to stimulate them. And so one could wear a device and then activate those ganglion cells in the right pattern. So maybe you have a device that kind of interprets the scene and stimulates them the correct way.
Thank you. I have another question here. You talked a little bit about those algae proteins, and Angelo's asking, where would you put the algae protein in the helper cell?
It goes into the membrane of the helper cell. So the virus contains the instructions for how to make it, the algae protein called a channel rhodopsin, and then that infects the neuron, and the neuron turns those instructions into the protein. The protein's put into the membrane where it can receive photons, where it can receive light, and then convert light into an electrical signal.
Thank you. And we have another question for you here from Bill. So Bill's saying, if the need for additional associated proteins is a limitation to the use of rod or cone opsins for optogenetics, why not simply add the codes for those proteins to the optogenetics transfection mix? And how does the nanoscope strategy get around this limitation? I love the use of simply in there. Like, that doesn't seem simple to me at all, but I'll let you answer that one.
I guess I can say a bit, but I have a feeling that Julie and Brian could say more. The kinds of viruses that we use in the laboratory and the ones that Brian mentioned beforehand have a limited capacity. They can't hold too much stuff. And so channel rhodopsin, the optogenetic channel, is small enough to fit into the virus, but the virus just can't carry all, the human opsin plus the 3 bits you need after that to make the whole sequence work. So that's what I think the short answer to that question is.
Yeah, that's a great, like Arjun said it perfectly. That's been a problem in traditional gene therapy too for large gene disorders that cause outer retinal dysfunction. So some of the genes that cause early onset retinal degeneration or Leber congenital amaurosis often involve very large genes, and it's difficult to deliver those very large structural genes in the viruses, what we call it, adeno-associated viruses that seem to be so effective with getting genes into the retina. And it's requiring new and creative genetic engineering approaches to change those genes. People have been looking to use CRISPR, for example, to try to edit genes instead of trying to deliver them back into the retina. Split gene approaches with splitting the gene product up and putting it in 2 different virus vectors and trying to get them into the same cell, and tackling the inefficiencies that come with that. So it's a problem that spans not, I think, just optogenetics, but retinal gene delivery in general. So.
Thank you both for that. Bill had follow-up here as well. He'd said all of the optogenetic therapies target cells in the pathway from rods and cones to the brain, bipolar and ganglion cells. So all of them fail to reproduce the contributions of circuitry in the OPL to the neural encoding of image details. And he said no one yet has mentioned Sheila Nierenberg and her strategy of using optogenetics to make retinal ganglion cells light responsive, and then using compound simulation of retinal image processing to stimulate them with light pulses. Does anyone have any thoughts on that particular work?
Arjun might have a few thoughts on this, but I think like everything, it comes down to what will be the best way to stimulate the optogenetic stimulation, the light patterns. And so one could go either of 2 ways. One, if you do introduce the opsin to the retinal ganglion cell, then what you're bypassing is the normal inputs that tell the retinal ganglion cell when and how to respond to light. And so then you have to use optogenetics to, I think, essentially, computational stimulate that cell. Practically speaking, it's not clear to me how that works. Perhaps Arjun knows, but that's one strategy. The other strategy is to deliver the opsin a little higher up, where you're using the native circuitry and the ability of those cells to, if their connections are maintained, then they're providing the balance of the patterns of excitation to the retinal ganglion cell, and then this retinal ganglion cell, because the inputs are still connected, it kind of reactivates. It will be reactivated in the proper way. I mean, they're both very challenging. They have their own challenges in terms of where do you focus, how do you then use optogenetic stimulation to stimulate those properties. I don't know if you have anything to add.
No, I think that was great. Yeah.
Wonderful, thank you. So then I have another question here from Denise. I think this is a question probably a lot of people have thought about today. So is it possible to determine if your ganglion cells will respond, or if you have enough of them to respond to optogenetics therapy? And then she said, is everyone who has RP, would they be a candidate for optogenetics therapy? Dr. Ballios, do you want to start?
Sure, we could talk about a little bit about how to assess ganglion cell function clinically. There are clinical electrophysiologic techniques to assess ganglion cell function. We use electroretinography most commonly, at least in my practice, to assess conditions that affect the outer retina, the light-sensitive photoreceptors. But there are ways of using electroretinography to assess the health and function of the ganglion cells. And so there are protocols that can be used to see how well those cells are making electrical signals. And so that's one way of assessing their function objectively. There are new imaging techniques to look at the structure of the retina and, and the thickness of those ganglion cell layers. So there's structural imaging and functional electrophysiologic techniques to tell us about the health of the ganglion cells in the context of patients who are also losing light-sensitive cells in other parts of the retina. We can't perhaps depend as much in these conditions on the information we get from the visual field, which we use a lot in glaucoma. We use visual fields to follow patient function. But if you have a patient who's also losing light-sensitive photoreceptors, then they're losing visual field because they're losing light-sensitive cells. And the visual field, which is being used to follow glaucoma patients, may not be able to really reflect how the ganglion cells are functioning because you're losing those light-sensitive cells. But there are other techniques we can use to assess their function.
And I think that the second part of that question, let's see, now I've lost the question, but it was like, is there a particular amount of cell function that needs to remain for optogenetics to be functional, to work? Or is that unknown at this time?
I think that's unknown, and I would say that's one of the project goals of this grant. So, perhaps we can assess the types and the proportions of retinal ganglion cells that are still functional, and we can certainly take further steps to then perhaps even directly manipulate in our experimental model system the number of retinal ganglion cells, and then ask what proportion is needed to get some kind of functional recovery in terms of functional assays or behavioral assays.
So stay tuned. Stay tuned.
I think these are testable, at least in the context with an animal model.
Okay, I think we have time for one more question. I think we have another question here about genetics. So David and Grace asked, they said, in a family of 11 children, 8 male, 3 female, the dad, oldest son, and the third son were diagnosed with RP. One of the sons has had genetic testing and they know the mutation. So they're asking, would it be safe to say that the father and the other son would also have the same genetic mutation?
I think the without knowing this family, I think and really hearing about their condition and how it progressed between the different patients in the family, I think yeah, a good first guess would be that it's probably the same causative mutation that's shared between the family members, with the little asterisks beside it, that expressivity of genes and the expressivity of that condition can vary between family members and across generations. And so it's not, you can't always predict by looking at one member of the family how other members of the family are going to experience their condition as years and decades of life go on. So usually when we identify when we find one person in the family and the first patient we see in the family who has the condition, we call them the proband. This is the person who has the condition, and that's the genetic term we use, call them the proband. This is the person where we identify the misspelling in the DNA. We then usually like to go to the other family members who we think may also have the condition or who are reporting symptoms and do genetic testing of them to verify that they also carry the same misspelling in the DNA. And that completes the picture for the family. But at first glance, yeah, it seems likely that misspelling is the one that's common across the family. And that's about all I could probably say with that information.
Absolutely. Well, thank you all of you so much for being here and for answering those very varied questions. And honestly, I think some of those questions that people have a far greater understanding of how the eye works than I do. So I really appreciate all of your insights and just, being here and being generous with your time. So